Yeah.
Why would you not call ATP a high energy molecule?
I call it a high energy molecule. ATP?
Yeah. So did you mean to circle ADP for energy?
No. Did I? I circled it for some other reason. These are the energy-rich guys here. These three. 1 and 2.
Makes sense.
Which ones are the electron transfer for ATP synthesis? Is that just any DH?
Any of these guys. So this one. If they're reduced cofactors and they're involved in the chain of electron transfer to ultimately make ATP. You don't worry about if that sounds big and confusing, don't worry about it, because we're going to spend weeks in here talking about it.
Okay.
Yeah. But I like to give a little overall context so you kind of see where we're going. Hopefully. Other questions? So. Other characteristics of glycolysis. As I mentioned, it's anaerobic. But cells that are anaerobic, and we've got them today, go down to the bottom of the Pacific Ocean and scoop up some sediment, and there are going to be organisms down there, and there ain't no oxygen to speak of, or very, very, very little. Right? And so they are running their home metabolisms. And you go in the landfill and dig down, more than a foot below the surface of the gunk, and it's full of organisms. In fact, you're evolving methane out of that landfill. Now we're trying to figure out how to capture it so we can burn it and not have to dig up so much. Not let it vent to the atmosphere where it's also a greenhouse gas. But the point is that there's bugs in there that are living lifestyles that have nothing to do with oxygen, right? And they have to make ATP, so they have no oxidative phosphorylation in the way that we think about it in this class. So they have some substrate level phosphorylation, but they don't have an electron transport chain to oxygen. Right? So in that big picture of the global cycle that I wrote, I was really writing, the main aspects of plants and heterotrophic cells, mainly, animals and other cells, other organisms. Right? But there's this whole anaerobic world, and that's where glycolysis originated. So the enzymes we're going to talk about are, billions of years old, so to speak. Or at least the template for them is billions of years old. And you can find hexokinase, the first enzyme in the pathway, in some primitive bug that phylogenetic dating tells us originated 2.5 billion years ago. And you can find hexokinase in your cells. And you can solve the X-ray structure of those two hexokinases. And they're not identical. They might be 50 amino acids different out of 200 or 300, or maybe even 100 different. Right? But they're going to be similar enough so that we basically say, yeah, they're the same enzyme. And to the extent they're different, it might reflect unique properties of what it's like to live in an anaerobic environment. Like maybe the KMs for the substrates are different because other aspects of metabolism are different. And so that's interesting. That becomes a window into the metabolic processes of the cell through that enzyme, which is the same in this ancient bacteria and humans. Okay, so it's anaerobic, it's ancient. LUCA means last universal common ancestor, which was some single cell that looked something like what we have today in the sense that it went DNA to RNA to protein at least, not an RNA world kind of thing, but a universal common ancestor of cells that had genetic material and coded protein synthesis and metabolism that was at least somewhat recognizable. And highly conserved enzymes in glycolysis. The enzymes are all soluble in the cytoplasm. So if we're talking about a eukaryotic cell, which often our context in the class will be a eukaryotic cell, then we want to be able to distinguish the compartments of the cell. We don't have to be cell biologists for this. We just need to know the names and basic functions of a few of these compartments. Right? And so eukaryotic cells have two plasma membranes around them. Right? There's a periplasmic space between the two, and then there's the inside, which we call the cytoplasm. Then, of course, there's a nucleus, which is a membrane-bounded organelle, and a mitochondria, which is a membrane-bounded organelle, and some other stuff. So all the glycolytic enzymes are insoluble in the cytoplasm. They're all soluble They're not, which means they're not membrane associated. So sometimes enzymes and cells are embedded in membranes, as you learned last term. Sometimes they're associated with membranes, like they're kind of stuck partly on there, not really embedded, but still associated. Glycolysis enzymes are all just soluble enzymes. And diffusible, meaning that they can move around in the cell, and they're not stuck somewhere. That's important because we'll come to distinctions where there are enzymes that are stuck somewhere. And so then the metabolites have to get to the enzymes rather than the enzymes perhaps to be using to find them. Right? And that aspect of things, you know, what's fixed and what's moving and how the, you know, where are the organelles in the cell and how are things moving around between them. That's an important part of metabolism. And it's not just about this enzyme and then this enzyme and then this enzyme and this mechanism. And, right? That's also an important part of it, but you know, we're going to try to put it all together by talking about all the parts of it, sometimes at the same time. Okay. Questions? So, glucose is the 6-carbon fuel molecule that is the starting point for glycolysis. Lots of cells, you know, we grow them on glucose in what we would call rich medium in the cell in the lab. But glucose has a lot of roles in the cell that are not necessarily just to go through glycolysis. So here's glycolysis, 10 steps. It's going to form a 3-carbon compound called pyruvate. In fact, it's going to form 2 pyruvates. Right? You might make note of that right now. It's like a crucial point. It's going to be on a bunch of other slides. But 1 glucose makes 2 pyruvates, pyruvate being 3 carbons. So what else can glucose do? It can also get oxidized a different way. This is oxidation. Right? This is also oxidation, but it's a completely different pathway for oxidation. Why would the cell want to have 2 different pathways to do the same thing? The answer is that it's not doing the same thing, in the sense that the byproducts of the oxidation of glucose are very different, and those have different roles in the cell. So we'll talk about this pentose phosphate pathway after the midterm. And then glucose is stored in cells as glycogen. Depending on the cell, it could also be stored as starch, sucrose, plants. So we're going to spend some time on those pathways. How you get to glycogen from glucose, how you get to glucose from glycogen, and how it's regulated. Because you can think about energy and the capacity to make energy this way, to make ATP this way. Right? Depends on how much of this there is around. And if cells are already in a happy energy-rich state, they don't need to go this way. They should probably be taking this glucose and going this way, making some glycogen out of it for the lean days, right? For the times when there isn't so much energy around, at which point we want to break that back down. Okay, so there's a very important regulation going here as well as here, right, which we will get to. And finally, you know, structural parts of the cell, sugars in the cell walls, etc. Right? And glycogen, glucose is a precursor. Not that glucose itself is in the cell walls, but that is a precursor for the synthesis of a bunch of other stuff that's in the cell walls. So, our first look at the pathway. It's a 10-step pathway, and you will need to be able to write out the structures of these compounds. That will not be the case for later pathways, but for this first one, I don't think there's any way to fully appreciate and get into the level of having good facility unless you go through the process at least once of having to write out a full pathway. So take a big stack of paper, write glucose on the top, and as we go through the reactions, you'll see that they're not going to be as hard to remember as you may imagine. Like what's the structure of the one at 0.7, right? But if you know how you get there, it'll be easier. So glucose you already know, right? And this is called the preparatory phase. And one thing we might notice about the preparatory phase is that all these intermediate metabolites, which are all stable molecules with certain, free energies, just like, all the there'll be thermodynamic conversions associated with each of these delta G0 primes. These are all phosphorylated, right? And in the second part, they're also all phosphorylated. Until the very end, here's your 3-carbon pyruvate. Notice it says 2 of them, because you got a 6-carbon glucose. This is what pyruvate looks like. And it's not phosphorylated. Right? But all the other ones here are also phosphorylated. Okay? So why would they all be phosphorylated? Why do cells want to? Why would it be adaptive for cells to have all their glycolytic intermediates phosphorylated?
So does it move out of cell?
So you can't, so they do not get out of the cell. So they don't have the capacity to diffuse through the membranes and get lost. That's a big reason why they're thought to be phosphorylated. Or at least at the very least it's a subsidiary function of having to be phosphorylated. Of course, phosphorylating them leads to the synthesis of compounds that are quite high energy. And so here in the second part of the pathway, you can see that here at step 7 and step 10, ATP is getting made, right? In fact, 2 ATP is getting made here and here, so 4 ATP are getting made from 1 glucose molecule, right? This pathway is written, these 2 slides is written from 1 glucose. So we're making 2 plus 2 ATPs, meaning, and these are the substrate level phosphorylations at step 7 and 10 to make ATP on the enzyme, kinase and kinase. Notice the name of the enzyme is a kinase, and we encountered kinase before when we were talking about post-translational modification. To phosphorylate serine, that would be a kinase enzyme. So here we've got a kinase that's phosphorylating ATP.